So our first lecture is basically just an overview of many topics we'll do in greater detail later, but we will start to talk about some experiments where people were trying to discover where these microorganisms came from. So once they finally knew they existed, the question is, do they just magically appear, or do they come from pre-existing microorganisms? So we will talk about that. At least we'll start talking about it and probably finish it up next time. So microorganisms are tiny. That's why they're called micro. And almost all of them are too small to be seen with the naked eye. So we need something to help us see them. What is that something?
The microscope.
Right, we use microscopes, and we'll start talking about those both in lecture and lab on Wednesday. The word germ is not a great word. It refers to a rapidly growing cell, but it actually refers to any kind of microorganism, and that includes viruses. So microorganisms do a lot of wonderful things for us. They decompose organic waste. So when a plant or animal dies, we say that it decomposes, but it does not decompose itself. Bacteria and fungi actually decompose that dead plant or animal. And when that happens, all the nutrients that make up that dead plant or animal are put back into the environment where they can be reassimilated by other living organisms. Microorganisms are also primary producers in an ecosystem. You may or may not have talked about this in 150, but when it comes to ecosystems, there are organisms that form the base of the food chain. And the most common example are plants, right? Because plants use sunlight to make their own food. And then you have animals, the herbivores, eat the plants. And then you have animals, the carnivores, eat the herbivores that eat the plants, and so on and so forth. But without plants, there are no animals. Well, many microorganisms also form the base of food chains. So that is one of their important roles. Microorganisms can also produce a variety of chemicals like ethanol, which I'm sure that most of you have no idea what ethanol is or what it tastes like because you're too young for that, right? But they can also make things like acetone, and I always tease the ladies about acetone because they love it. And why do you ladies love this toxic chemical?
Takes off our nail polish.
Right, your fingernail polish remover, right? So, but it is a toxic substance. We use microorganisms to help us produce foods, mostly through fermentation, like making things like vinegar, cheese, bread. None of that is possible without microorganisms. Now, in this course, our main focus will be on bacteria. So most of our time will be spent talking about bacteria. Our second major focus will be viruses. And we'll have a special lecture on COVID-19 this semester. We'll have a short lecture on fungi and a short lab where we look at some examples of fungi. In lab only, we'll talk a little bit about protozoa and see some examples, and we don't care about algae at all in this course. It's mostly bacteria and viruses. Now, we can also genetically modify bacteria to be factories for us. We can actually use them to make things like cellulase. And cellulase is an enzyme that breaks down cellulose. So we find cellulose in plant cell walls. And as you recall, cellulose is made of glucose. So there is tremendous potential energy in cellulose. The problem is animals cannot break the bonds between the glucoses in cellulose. there are many microorganisms that can. And so we can take a lot of, leftover plant matter. So, for example, corn. You've all seen fields of corn, right? And those big, huge, tall corn plants, they're 6 feet tall, and they typically produce 1 ear of corn. So that big, huge plant typically only has 1 ear of corn on it. So what do you do with the rest of the plant? Well, you can grind it up, mix it with cellulase, and more simple sugars will be produced. We can use those simple sugars, mix them with bacteria to help ferment them to make ethanol fuel. So we can actually get a lot of energy from the plant cell walls by using cellulase, and we can use it to make things like ethanol fuel. Animals that eat plants, and that's all they eat are plants, they have microorganisms that live in their gut, which produces cellulase to break down the cellulose, so then the animal can absorb the simple sugars and get energy from it. So without microorganisms, all these plants would not be able to be used as food for animals. When it comes to insulin, of course, we all need insulin. The problem with diabetics is either that they don't produce enough insulin or they don't produce any insulin at all. And so what diabetics used to take was animal insulin extracted from the pancreas of dead animals. And that was not human insulin, so that was not as good. What scientists eventually did is they found the gene for human insulin and they cloned that gene into bacteria. So now the bacteria produce human insulin. And then we just purify it from the bacterial culture. And that's much better for diabetics to use. So we can genetically modify bacteria to produce substances that we want. And of course, there are pathogens. And pathogens are disease-causing organisms. And although it seems like there are a lot of pathogens out there, in reality, they make up a fraction of 1% of all the microorganisms on the earth. Most of the microorganisms on the earth are beneficial to us in some way, and the rest don't hurt us and they don't help us, right? They're just there. And there's a very, very tiny number that are actually pathogenic relative to all the other microorganisms on the earth. But it just seems like there's a lot of them. So when human beings first discovered microorganisms, they were then able to study them because it's like, what do these tiny little things do? And when we learn that they are capable of doing things like spoiling food, we develop ways to prevent them from spoiling food, like refrigeration, like chemical preservatives. When it came to the spread of disease, we learned that things like washing our hands help to prevent the spread of disease. When it comes to aseptic technique, also called sterile technique, that's what you practice during surgery or what we do in the lab in micro. And the idea was to prevent the introduction of unwanted microorganisms, so to prevent contamination from occurring. Because prior to about the 1860s, humans, when they performed surgery, they didn't wash their hands. They didn't wash or disinfect or sterilize the instruments they were using because they didn't understand about how microorganisms could cause disease. So it's pretty scary when you think about it, but doctors never used to wash their hands right before performing a medical procedure. When it comes to naming and classifying microorganisms, we will talk in more detail about this later. But you may be familiar with the scientist Linnaeus, who came up with the system of nomenclature, where you call something by two names, the genus and the specific epithet or the species name. So we use the genus and species to refer to organisms. And who can give me an example of a genus and species? Come on, give me a genus and species. Like us, what are we? What are you?
Isn't it Homo sapiens?
Right, Homo sapiens, right? The genus Homo and the species or specific epithet sapiens. So that's how we refer to living organisms. Now, when it comes to writing a scientific name, of course, today we all have keyboards to type things, so you put those names in italics. In the old days, when you were writing by hand, you would underline the genus and species. And the genus was always capitalized and the species was lowercase. And these names are derived from Latin, or they're made to look like they came from Latin, so they're Latinized. And the thing about these names is that these are the same names used everywhere in the world. Now, locally, different countries will use their own names in their own languages, but if they're publishing a scientific paper, if they're presenting at a conference, we all use the exact same names which are derived from Latin. That way we all know we're talking about the exact same organism. And these names have meaning. They tell us something. They may tell us who discovered the organism, where the organism lives, what disease it causes, things like that. But whoever discovers a new organism gets to name it. And so, yes, scientists are nerds, so they've come up with some kind of, cute, funny names for things. But whoever discovers something gets to name it. So one example, Staphylococcus aureus. You've all heard of Staphylococcus aureus or Staph aureus, right? So staphylo refers to clusters. Coccus was shaped like a grape, so spherical. And the aureus, what is the symbol AU? What's the symbol AU? What element? Yes, Clarice, it's gold. Now, here's the thing. The scientist that was studying that particular strain of Staph aureus, that strain produced a golden yellow pigment. In reality, most strains of Staph aureus do not produce a golden yellow pigment. They're just kind of whitish or off-white in color. But like I said, whoever discovers it gets to name it. And that particular strain of Staph aureus did produce a golden yellow pigment. So that's why it's aureus. But in real life, usually you don't see any golden yellow pigment. Escherichia coli. That's why we say E. coli, because it's much easier, right? Named after the scientist who discovered it and where the organism lives. Now, when it comes to these big names, I do not count spelling. So spelling does not count. You don't have to be able to spell these big names, but you need to know them when you see them. Because most people do not like to write out these big long names, so they abbreviate like S. aureus, E. coli. You need to know what the S means, what the E means, because these letters don't mean the same thing. S could be Staphylococcus, it could be Streptococcus, it could be Serratia, it could be all different kinds of things. Same with the E. It could be Escherichia, it could be Enterobacter, it could be many different things. I expect you to know what those names are, but not be able to spell them, just know what they are. So the bacteria, they are prokaryotes. What is a prokaryote? What's special about it? Prokaryote. I know you learned that in 150.
It doesn't have DNA.
It doesn't have what?
DNA?
Oh, it does, but that DNA is not enclosed by a nucleus, right? Just like Sarah said, it's not enclosed by a nucleus. So there is no nucleus. There's DNA, but no nucleus. most prokaryotes do have cell walls made of peptidoglycan, and we'll learn a lot about that substance later on. And they mostly divide by binary fission, and we'll learn a lot about that later. We're also going to revisit one of your favorite topics in biology, which was cell respiration. I know how much you all love that stuff, right? So bacteria are very metabolically diverse. They can eat a wide variety of foods for energy. So we are going to review all the basics of cell respiration, but also focus specifically on bacteria and how they do things. We will mention photosynthesis, but we're not going to talk about it at all. And I know you're grateful for that. So viruses are the second major group of organisms, quote unquote, that we're going to talk about. Viruses are not cells. So they are not cellular organisms. What they are is a piece of nucleic acid, either DNA or RNA, but it's one or the other, not both. And that nucleic acid is surrounded by a protein coat. And there may or may not be a lipid membrane that surrounds them. But viruses are inanimate objects. They're not living things. They're just a piece of nucleic acid surrounded by protein. They can only do damage when they infect a host cell. And then the host cell gets forced into creating new viruses. But viruses have no independent metabolism, no independent replication. They cannot do anything by themselves. And yet they can do remarkable things when they're inside of a host. And we'll talk about that a lot later. So when it comes to classifying microorganisms, we'll talk more about this later on, but there are three domains of life, so three major groupings: the bacteria, the archaea, and the eukarya. The bacteria and the archaea are both prokaryotes. So the largest, groups of living things are prokaryotes. Yeah, Christiana? Nope. Okay. So two of the three domains of life are prokaryotic. And then eukarya has all the other living things that have a nucleus. So we have the protists, fungi, plants, and animals. So a very brief history of microbiology. So the first forms of life believed to have evolved were prokaryotic cells, right? The simplest cellular organisms. It was not until about 1673 when we were actually able to see them for the first time. And then once we knew about them and could see them and were able to start studying them, a bunch of scientists came up with the cell theory. So the cell theory is that living things are composed of at least a single cell. Cells come from pre-existing cells, and the cell is the smallest unit of life. And I put a question mark because this is a theory after all. And so that means it could change. It could be modified in the future. And the reason for this is because we may have to redefine what life is. So examples, there are some gigantic viruses called giruses for short, but these are enormous viruses. They are bigger than some bacterial cells, so they're huge. In fact, when scientists first saw them in the microscope, they just assumed there was some kind of bacteria and ignored them at first. When they went back and looked more closely to study them, they realized these were not cells, right? They were not cellular organisms. So they're actually viruses, but they're huge. And they have huge genomes, right? Which are bigger than some bacteria. They also carry genes that you only find in cellular organisms, not viruses. So the question it became, where do these giant viruses fit into the tree of life? Because they do possess some genes you only find in cells, but they're not cells, right? They're still not independent organisms. They need a host in order to reproduce and survive in. And right now, the primary host just seems to be amoeba, a type of protozoan. So we don't have to worry about these things, but they're huge. Some examples are the pithovirus, the mammavirus, mimivirus. They're huge, and some of them are shaggy, right? They're hairy looking. So these are very bizarre things. And so the question, became, where did they come from? What do they represent? Maybe these giant viruses represent degenerate cells. So say they're organisms that lost a bunch of stuff. Or maybe these giant viruses represent the evolutionary step towards cellularity. So it's possible that viruses or virus-like particles were the first things to evolve. And then those gave rise to the prokaryotes and then the eukaryotes and so on and so forth. So the cell theory is just a theory. It could change in the future. We may have to change the definition of life. But for right now, the smallest unit of life is at least one cell. But that could change. So now people were able to see these tiny little things, and the question became, where do they come from? There were two competing hypotheses. One was spontaneous generation, that these microorganisms just magically appeared from non-living matter. Or of course the other was biogenesis, life comes from life, right? Because humans have known for a long time how babies were made, and they've seen animals reproduce. So some people believe that these microorganisms, they have to exist first, and then they create other microorganisms. But others believe they just magically appeared. So we are going to talk about some of the experiments which were performed, and we're going to have to think about these experiments in two ways. We have to think like how the people 200 years ago thought, or even more than 200 years ago. And then we have to think in terms of what we know today, because we know a lot more today than they knew back then. So the results may mean two different things depending upon how you're thinking. Are you thinking like those people a long time ago, or are you thinking like today? So our first experiment involves maggots. So we all know what maggots are, right? The larvae of flies. So of course human beings had long since seen dead animal carcasses, and they're crawling with maggots. And so most people believed that the maggots appeared spontaneously from the dead animal carcass. They just magically appeared from that dead animal flesh, right? So what Reedy did in 1668 is he performed an experiment where he used some rotting meat and he put it into jars. Some of the jars were wide open and others had a very fine netting over them. So in the jars that were open, flies could go in, land on the meat. In the jars with the fine net, the flies could not get in. So after a period of time, the open jars had maggots crawling over the meat. But the jars with the fine net, no maggots. So where did the maggots come from? Spontaneous generation or biogenesis? So maggots, spontaneous or biogenesis? It's biogenesis, right? Because the flies were producing the maggots. And of course, it was kind of hard for the people to see what was going on, but the flies were actually laying eggs in the rotting meat, and the eggs hatched to form the maggots. So Reedy demonstrated that the maggots did not appear spontaneously, right? Flies had to be present, right, to create the maggots. So that's all well and good. Flies and maggots are big. You can see them. But what about these tiny invisible microorganisms? Where did they come from? So in 1745, John Needham performed an experiment. So he took some meat and he cooked it up to make a broth or to make a soup. And he may have cooked it in the flask, for maybe 10 or 15 minutes and then sealed the flask, or he cooked it in a pot and then poured the soup into the flask and then sealed it. But whichever way he did it, in a few days, the broth was all cloudy and stinky and full of microorganisms. So John Needham proclaimed what? Spontaneous generation or biogenesis? Right, Sarah, spontaneous generation. So because he cooked, right, the broth, thereby believing that he killed everything, and then he sealed it so nothing could get in, and yet microorganisms still grew. So he believed that he had demonstrated that microbes come from magic, right? They magically appear. And John Needham was very popular. He was very powerful. So many people believed him. But here's the problem. If he cooked the broth in a pot and then poured it in the flask and sealed it, the flask wasn't sterile, right? And if he only cooked the broth for 15 or 20 minutes, that's actually not long enough to kill all microorganisms. So John Needham's results actually demonstrated biogenesis because he screwed up. He either used an unsterile flask or he left it open too long, but he contaminated his broth. So although he believed that his results supported spontaneous generation, in reality, they supported biogenesis because he screwed up. So, but he was powerful. Lots of people believed him. So 20 years later, when Lazzaro Spallanzani did the exact same experiment and did it correctly, nobody believed Spallanzani. So Spallanzani cooked his broth in the flask at least 30 minutes and sealed the flask immediately, and nothing grew. So Spallanzani's results do demonstrate biogenesis. But all the spontaneous generation believers said, well, of course nothing grew because you sealed the flask, and one, no air could get in, and two, the magical vital force needed for spontaneous generation could not get in. And so that's why nothing grew. Of course, that was all nonsense, right? There is no magical vital force. And do all living things need oxygen? So do all living things need oxygen to live and grow? Actually, no, they don't.
You do?
Did you know? No. What do we call those microorganisms that do not need oxygen?
Anaerobic?
Right. Yep, anaerobic or anaerobes. So in fact, Spallanzani was correct. His results supported biogenesis. He cooked everything long enough to kill everything. He sealed it immediately. If there had been any anaerobes in there, they would have grown, but he killed them all by cooking. So Spallanzani was correct. But still, he was just some, weird Italian guy. No one cared about him. People still believed John Needham. Because as I said, John Needham was powerful. He was in the church too, so he had a lot of followers, and they believed in magical, mystical things. So nobody believed Spallanzani, even though he did everything right, and he was right. It took almost another 100 years before it was finally conclusively proven that microbes come from pre-existing microbes. And so next class, we'll continue talking about Louis Pasteur and how he repeated these experiments and how he finally was able to prove to people all these tiny little things do not magically appear. They come from other tiny little things. So, any questions on what we've done so far. I said we will finish this up next time and start the next lecture. So are we good with this? Yes. Okay, so I'm going to stop this recording.